Hi, my name is Yifan Cheng. I'm a professor of biochemistry and biophysics at the University of California, San Francisco. I'm also an investigator of Howard Hughes Medical Institute. And this is the second part of my talk today. And then here I'm going to mostly focus on the work related to my own laboratory, and particularly our efforts to using single particle cryo-EM to study indigo membrane proteins. And for a number of years that our efforts is trying to bring the single particle EM about membrane protein from a blobology level to the atomic resolution in a way that we try to bring something looks like this, which shows a blobs of a molecule, to something like this one, which has a secondary structure feature, and to finally to something looks like this, which has atomic details. So this is our overall goal, our overall mission of the lab. And in this talk, I'm going to take you through one of our recent work, which is illustrating the transition from a blobology to atomic resolution. So a major target of our laboratory is working on the ion channel called TRPVI. And this is a sensor for the capsaicin and noxious heat. And in this work, we have a collaboration with David Julius' lab back since around 2009. And David Julius' lab was the one that initially identified and discovered this TRPVI ion channel. And the TRPVI ion channel is an excitatory ion channel in the PAM pathway. It can be activated by a natural compound from capsaicin, which is an ingredient which makes your mouth feel hot when you eat chili peppers. And this channel can also be activated by noxious heat, by the temperature. When temperature rises above 43 degrees C, the channel will be activated. And below this threshold, the channel will remain inactive. So in a way that this channel is an integrator of different thermal and chemical signals. And nowadays, in my laboratory, we also use this channel as a model system for our methodology development work. Now, using structural biology of membrane protein is a particularly challenging area in structural biology. And over the last 10, 15 years, and tremendous progress has been made by X-ray crystallography of studying indigo membrane proteins. Particularly, the progress was made in the membrane protein production using overexpression system and developed a new detergent and new method for crystallizing membrane proteins. Now, Cryo-EM, as I mentioned also earlier, that has also been used by to study membrane protein, particularly in the early time that the electron crystallography has able to determine membrane protein structure to atomic resolution level. And in this case, which membrane protein was crystallized into a two-dimensional crystal, and still using crystallographic approach. Now, however, the bottleneck of any crystallographic approach is that you need to first form a very well-ordered or well-diffracted either 3D or 2D crystal and of sufficient size so that you can use crystallographic approach. Now, this is a major bottleneck of any crystallographic approach, that if you don't have a crystal, you don't have anything. So therefore, our goal is trying to use a single particle cryo-EM to study membrane protein. However, this is it's a promising approach, which demonstrated very early on in the 1990s by Joachim Frank lab and Elena Serasheva, that they look at using the single particle EM approach and to study membrane protein, and particularly in this case, the ryanodine receptor, to also at the low resolution level, by showing the correct structure shape, how this molecule looks like. And then around like 2009, 2010, the best single particle cryo-EM structure of membrane protein are these two structures. Either is the proton-driven ATP synthase and also the IP3 receptors, both around the nanometer resolution level. And it's in the resolution just about resolving the secondary structure feature about particularly the transmembrane helices. Now, back specific to the TRPVI ion channel, around this time, the resolution was still really limited to the blobology level at a much poor resolution. This is not because of the, this is mostly because of really the technology of single particle cryo-EM, particularly to study membrane protein, was very challenging, was very limited. So therefore, the resolution of number of these structures show the relatively low resolution. Now, around the same time, the crystallography approach of the trip channel can only resolve the determine the structure of a specific domain called anchoring repeats domain. This particular domain has been solved by the crystallography and the structure. Now, our goal is trying to, using single particle cryo-EM and without forming crystal, to determine high resolution structure of TRPVI ion channel to atomic resolution level so that we can understand the mechanism of how this channel is gated at atomic details. Now, this all started by making the protein, and this work was done actually carried out for a number of years by Hu Kao, a very talented postdoc from David Julius' lab. Now, he made this construct which is so-called minimal construct, in which he truncated a little bit of C-terminal and N-terminal domain, which predict to be flexible. So that this is mostly for better expression. And also, initially, this construct was designed for crystallization. So that the idea is to remove all this flexible domain so that it can facilitate better for crystallization. Now, this channel can be expressed in the hexel and using mammalian expression system. And then once the protein was produced and being purified, Hu was able to reconstitute this protein back into liposome, and then use a patch clamp to measure the activity of this protein, and showing that actually the purified protein has the same character as a wild-type protein. So that's already a tremendous progress for the making the protein. This is already actually quite a number of years' efforts into this making this protein. Then the next step is to putting this protein into electron microscope to look at it. So the TRPVI ion channel was first expressed in the mammalian expression system, and then solubilized and purified in the presence of detergent. Showing here is a side secretion chromatography of a detergent solubilized and purified TRPVI. It's a very nice and single peak, suggesting that the protein is in a monodisperse and in a good shape. Then the next thing we did is that we put this membrane protein into to visualize them in the negative stain EM, showing here that indeed the proteins are monodisperse. However, the 2D class averages of these proteins, these particles, showing that they are actually still conformational, actually quite heterogeneous. And this actually poses us a tremendous challenge for study to try to determine structure of this protein to high resolution structure. So, and another challenge imposed by this one is the presence of detergent, making it very hard for us to making thin layer of ice. And because the presence of detergent changes the surface property and surface tension, and making it much harder to make a thin layer and ice and then with protein embedded in it. And instead that we tried something else, that is using the amphipol. Amphipol is also a kind of amphipolyl molecules, different from detergent. This one was first used actually by Van Kuber's lab, and then to study a large membrane protein. And then in this way, that the idea is that it can remove the detergent. And amphipol in a way is similar as detergent, but different from detergent. You can actually put in the substitute detergent with amphipol and then run this protein through the side secretion chromatography using buffer complete without a detergent or amphipol. So therefore, it's very stable actually for membrane protein. So we tried this one with the TRPVI, and then you can see that after substituting detergent with amphipol, the protein also remains as a dispersed as a single peak. And then visualize this by a negative stain EM. Turned out that this one now, in the presence of amphipol, the protein remains not only monodispersed, but also much more in a better homogeneous conformation, showing here in the 2D class averages of this one. This facilitated us to have a much more homogeneous sample to further study them in using cryo-EM. Now, showing here on the left is a typical electron micrograph of frozen hydrated TRPVI in amphipol embedded in the thin layer of ice. Now, this image is recorded using a scintillator-based camera. On the right shows here is a Fourier transform of this image. Shows that the tone ring goes to almost to 8 angstrom. This was very exciting when we first got this image, because this type of data enabled us to getting a structure of TRPVI at about 8 angstrom resolution. At this resolution, we were finally able to resolve the transmembrane helices. And we say, showing this movie here is the 3D density map of TRPVI. You can see that we can nicely visualize the transmembrane helices, and these helices can be interpreted very similar as these tetrameric ion channels. Now, this was a tremendous excitement for us, that this was the first time we started visualize the membrane protein in the transmembrane helices by single particle cryo-EM. But this also around the same time that we also work out the technology of using direct electron detection camera to getting structure of a macromolecule complex to high resolution structure. Therefore, around this time, we immediately, instead of trying to publish this structure, we immediately started using the new technology to study the same thing and then try to aim to get a high resolution structure. Now here on the left is an electron micrograph of again this TRPVI embedded in the thin layer of ice. But this image now is recorded with this K2 camera in counting mode and after motion correction. Showing right here is a Fourier transform of this image. You can see that tone ring all the way goes to 3 angstrom, suggesting this image really has a quality could enable us to getting atomic resolution structure. Now from this type of data, we are finally getting structure of TRPVI at about 3.3, 3.4 angstrom resolution. You can see here at this density map that is showing that a really nice resolved side chain. Based on this density map, we were able to build an atomic model de novo based on the sequence of the structure. And just to illustrate how the camera technology enable us from a relative poor resolution level to high resolution level. Showing here on the left now is a 2D class averages of TRPVI images, TRPVI ion channel recorded by using the scintillator-based camera. You can see that we're also resolving the shape of the molecule. You can see the transmembrane domain and also the soluble domain. However, if we have the same sample now recorded with a K2 camera after motion correction, that you can see there is a great more detail of these images. Now this is the one enable us to getting this structure. As I showed in another talk earlier on, that we also now have a newer version of a motion correction called motion code 2, which we can use to further improve the resolution and correct both global and local motion, and then apply the new algorithm to this same data set, that we can see that we can get a sample to a significant, even further improved resolution. Actually, now we're actually able to improve this same data set, the resolution, to close to 3 angstrom. Now, from this structure, this work was actually done by two very talented postdocs in our laboratory, Hu Kao from Julius' lab and Fuliao from my laboratory. And both are faculty members now and starting their own independent lab. Now, back to the structure, that because we don't need to form a crystal, this enables us also to capture the structure in different conformation. Because single particle cryo-EM, we do not need to form crystal. Therefore, this enables us to using a ligand if we can trap the protein in different conformation using ligand, this will enable us to actually determine structure of the same protein in the different conformation. In this case here, showing here that we actually have the TRPVI ion channel determined 3 different conformations. Showing on the left is the structure of TRPVI apo structure without ligand, showing that the channel is in a closed conformation. Showing bottom here is the single channel recording corresponding to this in this conformation in the apo state, showing the channel is in a completely closed conformation. You can see that in the ion permeation pathway, the channel has 2 major restriction sites. One is up, one is in the bottom. The both are in a closed conformation, showing the channel is in a closed conformation. Now, in the middle here is the structure of TRPVI in complex with capsaicin, which is the ingredient that in the chili pepper make your mouth feel hot. Now, showing in the, you can see that the ion permeation pathway showing that the lower part of the channel is in the open conformation. And corresponding to the single channel recording shows that the channel is actually in the presence of capsaicin is a flicking back and forth between open and closed conformation. This suggests that the open of the lower gate by the capsaicin could allostatically destabilize the upper gate, and then making the upper gate also transition back and forth between the open and closed conformation. On the far right here is the structure of TRPVI in the presence of a capsaicin-like molecule binding in the bottom, and which you can see that the lower gate is an open conformation, but also in the outer pole there's a double knot toxin binding to the channel, which also open the channel open the channel. You can see that now the channel, both outer gate and the lower gate are both in the open conformation. The channel is in a completely open state. Now, single channel recording shows that the channel indeed is completely in the open conformation. This suggesting that different ligands acting on the channel have different effects on the either out pole or in the lower pole, and they're suggesting that the channel can integrate different signal input from different ligand binding. But for us, technically, we want to also move this one to a next level. And then, so one thing we want to see is how can we put the channel study the channel in the lipid bilayer environment. This is actually meaningful. If we look back to the structure of TRPVI, the density map of TRPVI in complex with a double knot toxin and a vanilloid compound bonding, that this structure also gives us the conformation of the whole structure. However, the resolution or the quality of the density map of this channel actually is unsatisfying, put it this way. If we compare this channel with the amphipols structure, we can immediately see that the channel actually when the channel in complex with double knot toxin, the resolution is relatively poor compared with the amphipols structure. Now, this is the same protein, and the structure was determined using the same approach. So the resolution, the poor resolution, is not because of the sample difference. It's not because of the sample difference, or it's not because of the difference in the technology, and it must come from other reasons why this resolution is poor. So one of the idea we're thinking is that because in this case the channel is as I mentioned earlier, is first purified in detergent and then substituted with amphipol. In this case, the lipid molecule are all removed during the sample preparation. Now, it is from early work, it is known that the double knot toxin binding to the channel is first require the presence of lipids. So remove the lipids, and then that may be the major reason of destabilize this complex, and then that leads to a relatively poor resolution. Now, the goal, therefore, is how to put this membrane protein back in the lipid bilayer environment, and it can still study the high resolution structure of them in the lipid environment. Now, traditionally, there are a number of different approaches. Again, that this can be started by electron crystallography, because membrane protein was reconstituted into the 2D crystal is in the presence of lipid. Therefore, in that way, that one can actually put in membrane protein back into the lipid environment, getting high resolution structure. However, this approach, again, requires the formation of a crystal. And if we don't have a crystal, you don't able to get a structure. So if we want to use a single particle EM approach, now we could not take this one. But using single particle EM approach, there's a method to put a membrane protein in the lipid environment, bilayer environment, is to using the so-called lipid nanodisc. Now, there are early works demonstrate that this approach is actually feasible to use in the lipid nanodisc. In both cases, the resolution they're able to get in was in the relatively limited resolution. Now, then using lipid nanodisc can also be actually not using it, but another approach is to put in membrane protein into liposome, and then try to look at the structure of membrane protein in the liposome. Now, this method was firstly actually was carried out, demonstrated by Hannah Saber's lab, and followed by Fred Sigward's lab, and then using this approach to studying membrane protein in the lipid environment. The resolution of this approach is still right now limited in the relatively around like a nanometer or sub-nanometer resolution, not able to get an atomic resolution yet. Now, one other approach is still using liposome and then approach reconstituting membrane protein into liposome, but taking electron tomography using subvolume averaging technique to try to obtain the resolution, try to obtain the 3D structure of a membrane protein in the lipid environment. The resolution of this approach currently is also limited around like a nanometer resolution. Now, for our laboratory, we saw that we want to try the approach of using lipid nanodisc. And then this work was carried out by a very talented joint graduate student, Yuan Gao from my laboratory and Julia's lab. Now, Yuan finally able to, after a lot of hard work, he was finally able to reconstitute TRPVI into lipid nanodisc. Showing here is a size occlusion chromatography. Showing in the middle here is the peak corresponding to the TRPVI reconstituted into the lipid nanodisc. Now, if we examine this sample by negative stain EM, one can see that they can indeed reconstitute into nanodisc. Yuan was able to use in 2 different types of scaffolding protein, which form nanodiscs of 2 different diameter. On the top here is the 2D class averages of a membrane protein in a relatively larger membrane scaffolding protein, or in other words, can be reconstituted in a relatively larger nanodisc. And the bottom here is a slightly smaller nanodisc. And you can see from these 2 images that the bottom one shows that the specimen, the actual particle are much more homogeneous in a way, and because there is not a big nanodisc floating around there. And the common wisdom would be saying that this bottom one, using this smaller nanodisc, will probably be better one to enable us to getting high resolution structure. However, our concern is that when you have a smaller nanodisc, it may impose certain constraint to the transmembrane domain to preventing it to undergo conformation change. So for that reason, we still choose to use the larger nanodisc, so that in this case, the membrane protein is really floating around inside a relatively large nanodisc without being constrained by the membrane scaffolding protein. Now, showing here again, this is a typical electron micrograph on the left of a TRPVI in the nanodisc, and recorded by the K2 camera operating in the counting mode. On the right side here is a Fourier transform showing this tone ring all the way go to 3 angstrom, indicating the quality of the images. And the bottom here is the 2D class averages of these images, showing high quality of this data. And from this data, Yuan was able to getting a structure of TRPVI in nanodisc together with the double toxin and a vanilloid compound bond. Showing here, you can see the large disc is from the nanodisc. The membrane protein is floating inside the disc here. And this blue density should mark the boundary of the membrane. This is from the head group of the lipid bilayer, lipid molecules that mark the boundary of the bilayer. And the blue density here shows here are actually specific bound lipids, that are specifically bound to the membrane protein, that they are very well resolved. We can model them. You can see a number of lipids in the outer leaflet and also in the inner leaflet. The resolution of this reconstruction is significantly improved. There are some density, for example, this particular one shown here in the mesh, we actually observed the same density in the structure determined from amphipole structure early on. The resolution there, however, was relatively poor. We could not model this density. Now with this improved density, we are now able to model this one as a phosphocytocholin. Now, more interestingly, whether the question is that whether nanodiscs actually can improve the resolution of a single particle reconstruction of a membrane protein. Now, this is not exactly the case. Now, here we actually have the 3 different structures of TRPVI, both in nanodisc and also in amphipole. On the left here is the amphipols structure, and in the middle here is a TRPVI with agonist bond. On the right here is the TRPVI with antagonist bond. You can see that in the middle one is the one really have the resolution improved quite significantly from about 3.8 angstrom to about 3 angstrom. On the left here in the amphipols structure, the 2 structures shows you the amphipols in nanodisc or in amphipole give us almost identical nominal resolution. showing that the structure actually, when we compare it to the looks almost identical. However, there are still a little bit of subtle differences, and then which shows that the structure of the nanodisc actually is a good thing for the membrane protein. Here, if we take out the most outer alpha helices, and then by comparing the density from the nanodisc and amphipole here, from nanodisc and amphipole here, and we can see that in the one side of this helices, by looking at the side chain density facing inside of the protein, that one can see that this side chain density looks almost identical. Even the side chain can be all positioned in almost identical way, and suggesting that the structure, there's no change there. However, if we look at the side chain facing lipid pore, we can see that in the nanodisc case, that this side chain density are significantly better resolved than the one in the amphipole, suggesting that even for the membrane protein that facing the lipid pore, that when the protein is in the lipid nanodisc, that this is still providing a better environment for structure determination. So this is actually suggesting us that the lipid nanodisc is probably a generally good platform to study membrane protein, and not necessarily that it will give you different conformation, but it probably most likely will give you a better environment for membrane protein to sit there, and then for better structure studies. Now, in here, we also visualize some very interesting feature. For example, we can now see that this toxin bonding to the channel is really stabilized by the presence of this lipid. You can see that this lipid has its head group forming an interaction with the channel protein, and its hydrophobic tail forms a hydrophobic interaction with the hydrophobic part of the toxin. Now, this sort of tripartite complex is probably required to really stabilize. It's critical for this whole complex to be in a stable conformation. Now, it's probably most likely is that this kind of stabilization and facilitated by the presence of lipid is the major reason that we're able to getting a better resolution of this particular conformation. Now, we also see that a very interesting thing is that, for example, you can see on the left is an unlinked structure showing two lipids molecule. Its head group is more or less in a similar level, height level. And when the toxin binding here, it partitions into the bilayer, it will actually move the lipids up and down to accommodate the binding of the toxin. This is also quite reasonable to understand. Now, the further thing which is quite interesting is that we now have a much better resolution compared with previously in this vanilloid binding pocket. In the APO structure, we see a very clearly defined density map, which can be modeled very nicely by the phosphatidyl inositide molecules. And this can be, and we actually see this same density in the amphipole structure, but not so clearly resolved. So suggesting that this molecule, this lipid molecule, is actually mostly likely is an endogenous one, but not added during the purification or nanodisc reconstitution. Now, in the same pocket, in the agonist bond structure with the resiniferatoxin, which is a very potent agonist, TRPVI agonist, then we can actually see also improve the density. We can now model this one, agonist, how it binds there, and how it's induce a very subtle conformation change to the channel, and to understand how this agonist is actually acting on the channel. Now together, if we're putting all these things together, we can actually have a much better understanding of how vanilloid acting on the channel. Showing here, you can on the left side you can see is that the density of a vanilloid actually overlap with the density of this lipid molecule, suggesting that the binding of agonist displaces this lipid molecule. And such binding of agonist promoting a salt bridge formation, which moves the S6 helices backwards, and which in turn also moves S5 helices backwards, which coupled to the S6 helices also move backwards and leads to channel to the open. And this is how the agonist actions on the channel. Now, in the presence for antagonist, and this is the structure of a TRPVI with capsaicin, which is a very potent antagonist, we can see that this antagonist molecule also binding to the same vanilloid binding pocket. It also displaced this lipid molecule, which originally occupied in this pocket. And however, this binding does not promoting induce any conformation change in the channel, and then suggesting that this is the antagonist binding is probably stabilize the channel in actually a close conformation, and then providing the mechanism of how the antagonist is working. So this actually gave us an idea about how channel is a further more detailed mechanistic information about how agonists and antagonists are acting on the channel. Now back to the general question about how membrane protein can we study membrane protein in a lipid environment. As we demonstrated here, the membrane protein can be put into lipid nanodisc to study the structure of them, to facilitating the structural determination of a membrane protein in a lipid environment. Now, this is our structure showing here of TRPVI in a lipid nanodisc. On the left here is a ryanodine receptor, which has a very large soluble domain, also embedded in the lipid nanodisc. In both cases, one can think that the structure, the image refinement was really facilitated probably by this large or relatively large soluble domain. Because in the transmembrane domain, we have a large chunk of unstructured mass that added to protein, which would, on the other hand, influence or interference with image alignment. And therefore, it seems like that to getting the structure of high-resolution structure of a membrane protein in lipid nanodisc do require certain size of soluble domain. And this imposes a question about whether how do we study a membrane protein which does not have any soluble domain? And can we still using the lipid nanodisc approach to study such membrane proteins? And then so there are a number of different ways that we can think about it. But in our laboratory for a number of years, we have been taking this approach to artificially increase the soluble domain by using a Fab, monoclonal Fab, particularly using a conformational specific monoclonal Fab that to bind to the indigo membrane protein. We can use this one as a fiducial marker to facilitate the image alignment. Showing here is an example of such a case. Now this is a membrane protein of a ABC transporter. It's a bacterial ABC transporter. It's a heterodimer, and the two monomers are very similar. So therefore, this protein actually has a pseudo twofold symmetry without actual precise twofold symmetry, but a pseudo twofold symmetry. And in this case, it does have some soluble domain, but because the two are very similar domains that may interfere with the image alignment, we could not distinguish which one is which at low resolution. So that's what's the concern. So in this case, we're generating a monoclonal Fab by using phage display library approach that panning of a membrane protein generating this specific conformation-specific monoclonal Fab, which binds to one specific monomer. And using this Fab as a fiducial marker, we were able to get in the structure of this one, membrane protein, to a sub-nanometer resolution. I have to say that this work was actually done in the earlier time when the K2 camera just became available. This is one of the reasons that this resolution was still in the sub-nanometer resolution, but not higher. But nevertheless, this actually demonstrated an approach that is using monoclonal Fab as an approach to study indigo membrane protein, and particularly to study membrane protein without a big soluble domain. Now, to finally make a conclusion here, that we're now able to demonstrate that actually also show that the single particle cryo-EM, using single particle cryo-EM to study membrane protein, actually has become a versatile or very powerful technique, particularly for membrane protein, for ion channel, for a large group of membrane protein that are particularly difficult for crystallization. And then for those membrane protein, nowadays that it is probably a more efficient way and then to study them to obtain a high resolution structure by using single particle cryo-EM. We also demonstrated that it is possible now to put in membrane protein into lipid bilayer environment using a lipid nanodisc approach to getting high resolution structure. And then also specifically to visualize the membrane lipid interactions, which may be better preserved in the presence of lipid environment here. And then in the last, we also showed that using FAB approach, that we were able to most likely to study membrane protein that with very small or even no soluble domain. And we hope that using FAB could be also another general approach that are able to enable us to study such membrane protein. And finally, I want to conclude this one to acknowledge people who contributed work. These are showing here, listed here, our current lab member and a former lab member. And in the TRPVI work, we have a very tight and close collaboration with David Julius' lab. And these are the people from Julius' lab, which also contributed to this work I presented today. And also that we have also a collaboration with Charlie Craig lab and Bob Stroud lab at UCSF to develop this technique using FAB to facilitate single particle cryo-EM study of small membrane protein. Finally, these are a funding agency which support our work. Finally, I want to thank you for your attention.